Hello and welcome to proteins. Right, proteins, amino acids. Know your amino acid structure. Um, so remember we've got our carboxyl group, and we've got our amino group or amine group. Biology, you can call it an amino group. This is from the syllabus. R means residue. It's the rest of the molecule. So this is the residual group, and it's different in the 20 amino acids. We'll have a look at a few of them. And then this is just a hydrogen to make up the bonds. When you ever drawing these out, remember, check bond numbers. Again, it's hard to kind of see here, but I've got my extended versions down here. So that's an amino acid. Let's join them together and form a dipeptide. So sometimes these are like written as like R1 and R2 to show that they're two different R groups, two different amino acids. Bring them together. They always have to be this way around. It always has to be a carboxyl group. A carboxyl group meeting an amino group, and we just bring it together, find water. So there's water, H2O, and therefore I can lose water, and we join up what's left. So that means I've got a bond between the carbon and the nitrogen forms. That's got a name. I've still got my double bond oxygen. I'll pop it down here. You can stick up or down, it doesn't matter. It tends to show them being opposite, but that new bond here, find a pen that's different, let's make it red. So this bond here, that's our peptide bond. Names of bonds get marks. Okay, to do this process, you need certain things. You need an enzyme. Actually, inside the ribosome is the enzyme. And you also need the input of energy in the form of ATP. Not directly, you need the ATP to load an amino acid onto a tRNA. Then there's a high energy bond that then releases that energy at the ribosome. But yeah, the peptide bond ultimately did get its energy from ATP. So that's a dipeptide that we've formed here. So on the pen, yeah, so this is our dipeptide. It's not a dipeptide bond, it's just a dipeptide. Two amino acids joined by a peptide bond. If you do chemistry, you'll do more about like the rotations of all these things, and you can get different like versions due to different rotations, etc. So that's that. Then I thought we'd just have a quick look at some amino acids. They often ask you to like circle the R group. So I've gone for leucine, isoleucine, and cysteine. I picked leucine and isoleucine just because, as the names suggest, isoleucine is an isomer of leucine, as in they have the same chemical formula. However, they're just arranged differently. The CH3 is on this carbon on isoleucine, whereas it's shuffled down one on leucine, but they behave differently. So let's just circle the R group. Yeah, so there it is on that one, and I've done them all. They're all the same. So it's dead easy. Really, it's too easy. Yeah, and there's our R group, and this is just to show that this one's one of the few ones that's got sulfur in its R group. Obviously, proteins are CHONs. Yeah, those are the elements in proteins: carbon, hydrogen, oxygen. They all have nitrogen, and then some of them have got sulfur in their R groups. And those that have sulfur can sometimes form disulfide cross bridges. So let's have a look at different levels of protein structure. So primary structure, this is just the sequence of amino acids. So that's a bit interest to like geneticists. Yes, it's interest to genetics. It ties in with topic 4, where you can compare the amino acid sequence, the primary structure of proteins, to get an understanding of genetic differences. Okay, so yeah, so there'll be people who will like tweak an amino acid to get a protein with a different kind of ability. So, for example, if you've got an enzyme, but it denatures at, say, like quickly at like 40 degrees C, and I want it in my washing machine, and I want to do a nice hot wash to get the whites whiter, and I don't want my enzymes to denature, then maybe you could like replace some amino acids with those that contain sulfur and get a more heat stable enzyme that I can take up to 90 degrees C. They do it in industry as well, so I've got the same active site, but it's now got like disulfide cross bridges holding its active site, and therefore you can make more of a product from an enzyme control reaction. Anyway, I've got off on one, but yeah, primary structure is just the sequence of amino acids. Secondary structure, again, this is due to, again, names of bonds get marks due to hydrogen bonds. Write hydrogen in exam, yeah, and these form between the carboxyl and amino group, so not the R group. And these form two levels of structure. You either get an alpha helix or you get a beta pleated sheet. So, for example, trying to think of one. Beta pleated sheets. Silk is beta pleated sheets. Alpha helices, things like collagen, a bit alpha helices. And often they'll show you like some kind of like 3D representation. If you go on the internet and look for like a protein structure, it will show like bits of alpha helix, and then a bit of like nondescript, and then a bit of beta pleated sheet that then goes into another little bit of alpha helix, that then goes into another bit of beta pleated sheet, and then goes into another bit of nondescript. So they kind of show the proteins by these kind of models. So you don't have to have, within the protein, there can be regions of alpha helices. Some proteins are entirely alpha helices, like collagen. Some proteins are entirely beta pleated sheet, like spider silk. I always imagine it being like boingy, as it's stretchy because of the beta pleated sheet. However, in like enzymes, there'll be a bit of alpha helix, a bit of primary structure that then hasn't got any kind of like particular shape, and then there'll be a region of beta pleated sheet to give this like complex 3D shape that we get onto when we get into tertiary structure. So if we form additional bonds between R groups, so this is due to, well, it's hydrogen bonds again. Hydrogen bonds, ionic bonds. They're the bonds that kind of break more with pH change. Hydrogen bonds break really easily with temperature. Ionic bonds, and we get our disulfide cross bridges. Disulfide cross bridges, is that pH? I never know anymore. Cross bridges. And yeah, between different R groups. Yeah, so the sequence of the amino acids determines the shape of the protein because of these R group bonds that form. And yeah, hydrogen, that again very easily broken with temperature. Ionic bonds that are more pH, and disulfide bonds don't really break. If you have your hair permed, perming solution breaks disulfide cross bridges. You then set the hair in the style that you want, and then you neutralize the chemical that breaks the disulfide cross bridges, and then disulfide cross bridges form in the new position where you position the hair. Hence, the hair has been permanently styled, and it would have to grow out. You've had a perm. Hydrogen bonding is the stuff that breaks with just a bit of heat from hair straighteners or a hair dryer, and then you just kind of encourage the hair to go into a new shape, and hydrogen bonds form in new positions. Yeah, it's all about hairdressing practically. Is proteins. Ask your stylist next time you're in there about bonding and their understanding of that. So that's tertiary structure, and then we get onto quaternary structure, and this is the one that gets a bit confused. But yeah, this is when there's multiple proteins joined together. Yeah, and joined is poor. Yeah, it's bonded together. Yeah, so the one I've drawn here hasn't got quaternary structure. It's one protein that could unravel it and form one strand. So this is where there are multiple proteins joined together. Examples that we know about that I put below are antibodies, with these 4 polypeptide chains joined together. We've also got hemoglobin, again 4 that are stuck together. I think DNA polymerase is technically got quaternary structure, but it's not one that's really we meet on the syllabus apart from what the enzyme does. So yeah, so just if you're describing levels of protein structure, yeah, you'd again you always start at primary, secondary, tertiary, quaternary. It's a bit controversial about does something like collagen have tertiary structure, because it's collagen's made of alpha helices that are then bonded together, but it's not yeah, hasn't got like a complex 3D shape like most tertiary proteins have. So it's a bit controversial. Again, it's not mentioned on the syllabus anyway. It's collagen, so it's best just to ignore that. If you go on the internet, you'll find like reasons for and against it. I think technically it probably does, because the R group bonds are involved somewhere, therefore it counts as. But anyway, yeah, proteins on the syllabus. There's loads of enzymes that you need to know. All the digestive ones. Just as a tip, if you're ever learning, like some enzymes, I always like the exo and endo and dipeptidases. They're a nice like group of them. DNA polymerase from year 12, DNA helicase. Again, learn your enzymes and learn the bonds that they hydrolyze or form. So exopeptidases, all these peptidases, they hydrolyze peptide bonds. DNA helicase breaks hydrogen bonds. It doesn't hydrolyze them. It just breaks them like a wedge. And then DNA polymerase, that's the phosphodester bond forming enzyme. Hemoglobin. Remember, this is made of 4 polypeptides, but plus they have this heme groups, and there, how they when oxygen binds, it changes the tertiary structure of each of those 4 polypeptides. So again, it's a really nice example of one where something binds and causes a shape change. Similarly for enzymes, I will do a lesson all on enzymes, because it's there's lots to that. Antibodies, again, 4 polypeptides. This is where we talk about the antigen antibody complex, or vice versa. It's a good example because it has disulfide cross bridges to hold them together. And you can imagine that if you're an antibody, so you're like a Y-shaped. So there's 2 of the polypeptides, there's the other 2 of the polypeptides. If you held onto a bacterium on that side, virus antigens, and one on this side, virus antigens, and they have a flagellum, and they're trying to swim away from each other, then you obviously need really kind of strong disulfide cross bridges within this to stop the actual protein from just being ripped apart. So it's quite a nice example of why disulfide cross bridges are important if you ever have to give one. Receptors, I just wanted to bring in because many things can be classed as receptors. So a cell can be a receptor. We have light receptors in our eye. We have touch receptors in our skin. You do about them in one of the topics in year 13. However, receptors can also be it can be a protein in the cell membrane, or even a protein within the cell if it's a chemical that gets into the cell, like estrogen does. So, if I was drawing a receptor, there's my phospholipid bilayer, and then in here we've got some kind of protein that's got like it looks a bit like an enzyme. It's got a certain kind of shape. Something's going to come and fit into that receptor, and normally you have some kind of like second messenger that it will like release. So as soon as the blue dot gets into the receptor, it'll cause a shape change on the other side of the receptor, and this green thing will be released within the cell that then goes and sends a message. So you learn this at GCSE with like neurotransmitters at the synapse. You know, they're released from one cell like a sensory neuron, and then they diffuse across the gap, and they stimulate receptors in like a relay neuron, and then this will trigger a new nerve impulse. So yeah, just receptors are can be cells and can also be proteins, but yeah, you'll meet them in a later unit. Transport proteins, topic 2, which can be things like protein channels. Yeah, so we get channels, and we get pores as well. Channels and pores. You get pumps as well. I would just call them all transport proteins. And if they require extra energy, if they require any extra energy, then you can bring in active transport to function. If they don't, so that's plus ATP, and if they just work on something bonding, then they're into facilitated diffusion. Again, it's probably another lesson, is all that transport anyway. But yeah, transport proteins, again, embedded in the cell membrane. And my favourite, and the one that's very much left, I always think, are histones. You meet them in topic 4, well, a bit in topic 2 as well, but you meet them a lot in topic 8, where we have our DNA. Here's our DNA, and here's a load of histone proteins, and the DNA spools around them. So you end up with this like spooling of DNA around histones. And any of the genes in the DNA that are associated with histones would be turned off because DNA polymerase wouldn't be able to get to them. So yeah, histones is a really interesting protein. It's how we control gene expression. I'll put that there. It's so important in controlling gene expression. Yeah, turning on and turning off genes. And then lastly is the test for proteins, and it's just the Biuret test. And you need a before colour and after colour. Well, it's light blue is the solution, and the positive test means it turns to purple or lilac. Mauve might be mentioned, but yeah, just make sure that you know your light blue and purple colour. And often they'll ask you, describe a test for protein. Sorry, describe a test for enzymes. Well, it's still enzymes are proteins, therefore it's the Biuret test. Describe a test for antibodies. All right, antibodies are proteins. It's the Biuret test. Done. 18 minutes. Longer than I thought that. I thought that'd be really quick, but I've obviously prattled on, so I do apologise.